All right, so we're presenting on what drugs could treat COVID-19.
So, hi everyone. As our wonderful host mentioned, I'm Lindsey. I'm a grad student at Rockefeller University, and I'm originally from Connecticut, which is that star that's a little bit to the left in red.
Yeah, and I'm Emily Costa. I'm studying at Weill Cornell, and I'm originally from Massachusetts, so that slightly shifted star, you're right, but it works.
All right, so I'm actually, when obviously there's not a pandemic happening, I'm super interested in understanding how behavior works, and specifically how the brain is able to generate sometimes super cool and sometimes not so cool behaviors. And so I do this by studying ants in Daniel Cronauer's lab at Rockefeller. And as many of you might know, ants are actually a really great system to study social behavior because they live in colonies and they communicate with each other, and they have division of labor. So some ants are specialized nursers and take care of the young, and some specialize in foraging and look for food, and they all kind of work together to keep the colony alive and happy. And my research specifically is focused on understanding how ants, which have, I mean, most of you have probably seen an ant before, they have very small heads and very small brains to go inside those heads, but they're still capable of these pretty extraordinary social behaviors. And so actually on the left here, I have a picture of an ant brain. And what my research is focused on is figuring out what brain pathways are important for social behavior and specifically communication between different ants to coordinate their behavior. But I'm not in the lab all the time. And when I'm not in the lab, I really enjoy spending time with my family. You can see them on the bottom left here. I think some of them might be watching. But I grew up in Connecticut, and so it's really nice. I can just hop on the train and go home and visit them. And I also really, really love animals. You can see our family's dog was in our family portrait. I have 2 cats. You might see them like running around on the cat tree. I asked them to stay as calm as possible, but they don't really speak human, so we'll see. I also love baking. I think it's a really nice way of sort of being creative and experimenting. It's especially nice at times like now where I can't be in the lab. And finally, I really love talking about science and thinking about new ways to communicate science. So I love volunteering with Rocky to You. I also have done other outreach sorts of things, and specifically one that was really fun was working with artists with intellectual and developmental disabilities to create bioart.
Hi, so I'm Emily, or Em for short, and I'm currently doing my PhD or graduate school, or I'm currently in graduate school at Weill Cornell, but I actually do my thesis research just down the street over at Sloan Kettering in Charlie Rudin's lab, and there I study a type of lung cancer called small cell lung cancer, and in particular, I'm trying to understand how to make immunotherapy work better for patients with small cell lung cancer. So immunotherapy is basically a type of cancer therapy that engages our body's immune defenses to attack cancer cells. So the kind of immunotherapy that I in particular study is called immune checkpoint blockade. And the way it works is basically there's this interaction that happens between tumor cells and immune cells involving 2 proteins, PD-L1, which is on the tumor cell, and PD-1, which is on the immune cell. And normally these 2 proteins interact actually as a function of preventing autoimmunity, which is when our immune cells accidentally attack our body's normal healthy cells. But tumor cells actually use this mechanism as a way to hide themselves from the immune system, which would otherwise actually notice that they're really abnormal compared to our healthy cells because they have so many mutations. So basically, we figured out as a therapy that if you block this interaction using antibodies, which can either bind to PD-L1 or PD-1 and keep them from binding to each other, then you can actually have the immune cells, specifically T cells, notice the cancer cells. So they can no longer hide from the immune system, and then they can attack and kill those cancer cells and start healing patients' tumors. So it's a pretty cool technique. And as I mentioned, my work is focused on using this kind of therapy and making it more effective in patients with small cell lung cancer. So when I'm not in the lab doing science, you can find me talking about science pretty much anywhere else. So I really love volunteering with RockEDU, both in their lab experience program and also at Science Saturday, which is a program they do every summer. This was from the last Science Saturday, where myself and a couple of friends in grad school ground up a lot of strawberries and then showed some kids how to extract DNA from them, which was a really fun, messy time. I also talk about trivia and share science stories on a podcast called Fax Machine that I run with some of my friends from grad school. It's funny and punny, and we have a lot of fun doing it. And I also really enjoy projects that kind of look at the intersection of art and science. So for example, last year I had the really cool opportunity to work on a project scripting a series of short films about the history of women in science. And I also enjoy doing this kind of photography called cyanotyping that folks in the 19th century actually used to use to kind of catalog botanical specimens and sort of like capture images of plants. Now I do it because it's just kind of a really pretty thing to do and a nice hobby, but that history is kind of cool, I think. All right, so the paper that we're going to talk about today is an example of scientific literature. But what do we mean by scientific literature? So literature, as we call it in the research world, is how scientists basically write about their research and then communicate it to each other in a way that's informative, expedient, and trustworthy. So as you can imagine, the first step in generating scientific literature is actually doing the research. So scientists will be in the labs, running experiments, collecting data, and then once they decide they have research they'd like to share, then they'll write about it in a paper. And then this paper will go to two places. It can go to an open source publisher. A really popular one in the life sciences is called biorXiv. And papers that go open source are available for anyone to read immediately, which is really helpful when the information that the authors are sharing can be used by other researchers really quickly, like COVID-19 research. It's especially useful to have open source journals. But a caveat to this is that the papers that go to open source publishers are not peer reviewed. Conversely, another place that papers can go is to peer-reviewed journals. So you've probably heard of some of these, like Nature, Science, Cell, PLOS, PNAS. There are loads and loads of them. But here, the papers actually do get peer-reviewed, and then once they're published by the paper, then they can be read by people in the scientific and medical community who will then apply these findings towards their own research. So I want to take a little bit more time, though, to kind of explore the differences between open source publishing and peer-reviewed journals. So to kind of talk about peer review and explain what that actually means, basically when authors submit a paper to a journal, the editor at that journal will then forward that paper to other experts in the field that the paper comes from. And those experts who are also scientists will look at the methods and the data in the paper and basically evaluate whether the experiments were rigorous or whether more experiments need to happen in order for the authors to actually make the claim that they make in the paper. So this process can involve the authors going back, doing more experiments, resubmitting to the journal, maybe getting rejected by the journal and then submitting to another one and starting peer review at that journal. So although peer-reviewed papers are regarded with more confidence than open source because they have this kind of vetting process that's built in, it also takes a lot more time for papers that undergo peer review to actually be put out where people can read them. Another main difference between open source publishers and peer-reviewed journals is the degree of access that people have to these papers. So some journals actually require that scientists pay them to publish their papers, which can then limit where and whether labs can publish. And additionally, some journals also require that you pay a fee or have a subscription to read the papers in them, which can then, as you imagine, also limit who gets to read those papers. A lot of journals nowadays are moving more towards an open access model where you can actually read their stuff for free. But overall, that whole situation is avoided with open source publishers. And one more kind of limitation to access that plagues scientific literature in general, no matter where it goes, is jargon. So scientists write usually using a lot of jargon, which are basically technical terms that are kind of difficult to digest if you're not a specialist or not in the fields that the terms come from, which can make the literature difficult to access for people who are not in the field, but it's also why we do data for the people. So there are ways to work around it. There are also, I should say, various types of scientific literature. So the paper that I was just describing and the paper that we're going to look at today are both examples of original research. So I mentioned these present the research, the author's hypotheses, their methods, and their data. But there are also short reports and letters, which kind of tell shorter research stories, review articles, which kind of summarize the salient findings in a single topic or field, case studies, which you see a lot in medical journals, and those will cover specific instances of interesting phenomena, like a patient who has unusual symptoms for a certain disease. And there are also methods papers where authors can showcase a new method or technique that they've developed.
All right, so moving into the paper that we are going to be talking about today, this paper is called A SARS-CoV-2 Protein Interaction Map Reveals Targets for Drug Repurposing, which is a lot of words. To break it down a little bit, in this paper, they're going to start by looking for protein interactions for the virus SARS-CoV-2, which we all know is the novel coronavirus, which causes COVID-19 disease. And we'll talk more about what protein interaction means exactly. And then after that, they're going to look for drugs that might be able to be repurposed or used in a different way to treat SARS-CoV-2. And we'll talk a lot more about this, and this is a super, super exciting paper, but it's also still very much the only beginning of research. And there's a ways to go before drugs like this, or drugs from this paper, could be used to treat patients. Another cool thing, and one of the reasons why we wanted to talk about this paper, is because of how fast, sorry, can you go back for 1 second?
Oh, sorry.
No, you're fine, you're fine. Is how fast the research has progressed. So you can see, or maybe you can't see, but this is a peer-reviewed journal article now. But when we started talking about making this presentation, like a week and a half ago, it was still a preprint. And one of the reasons why we think that this has progressed so quickly is because of how many authors there are on the paper. So there are, can you go to the next slide, please? Thank you. There are 25 authors, and they work in over 20 labs across 8 institutions, which is a wildly big collaboration. You can see those 8 institutions on the map. The stars are indicative of them. And you can see they're mostly located in the US, but there are also some European labs. And I think it's just been really cool to see how the scientific community has really come together to try to understand SARS-CoV-2 and how to potentially treat it. Also really cool about this paper, even though it's just come out, it's already been highlighted in the news. And so its findings have been discussed in the New York Times as well as in the conversation. So now we're going to turn things over and start talking about the body of the paper.
Great. So before we jump in, we'd actually like to take a little bit of time to sort of explore some key terms that have important roles in the paper, but that you might be unfamiliar with. So those terms are protein-protein interactions, viral tropism, phases of drug development, sort of conceptually, and mass spectrometry. So protein-protein interactions. So if you think of our cells as factories, the proteins are the workers. They're the ones carrying out all the various functions that our cells do to basically keep our bodies working and healthy and doing what they need to do. And a big part of how proteins do their particular jobs is by physically interacting with each other. So this works at sort of like 3D surfaces of proteins that are complementary. So if you imagine puzzle pieces sticking together, that's exactly how this works. And basically, once these complementary surfaces find each other, they then get held together by chemical bonds that form in between them. So in our cells, proteins physically interact to enable certain kinds of jobs. For example, some jobs can only take place if the proteins are together in a complex that are enabled by these interactions. Sometimes these interactions can allow one protein to make a chemical change to another one that then affects the activity of that other protein. Interactions can also allow proteins to move around the cell to different places to do specialized jobs in those places. And where to go? And protein interactions can assist or prevent other protein interactions that might otherwise be possible. So a very important thing to know too is that lots of drugs work by blocking disease-related protein-protein interactions. And in our paper, the authors are not only looking for protein-protein interactions, but also drugs that can disrupt them. So another term that we wanted to take a look at was viral tropism. So viral tropism is the ability of a virus to infect a specific host. And this can happen at various levels of biology. For example, at the cellular level, we know that HIV can infect macrophages, which is a kind of immune cell, but it doesn't infect neurons, which are cells in our brains. The flu virus infects our lung tissue, but not our brain tissue. And even at the whole organism level, myxoma is an example of a virus that infects rabbits, but doesn't infect us humans. And basically what determines whether a virus can infect a specific host is the presence of certain things called host factors. So these are cellular resources that a virus hijacks in order to infect and replicate within its host. So for SARS-CoV-2, which we know can infect bats, monkeys, humans, and maybe some house pets. We know that a host factor that it relies upon is the ACE2 receptor, which is a protein that is expressed in our cells that SARS-CoV-2 actually uses to enter our cells during infection. So the authors of our paper are looking for more host factors, essentially, like ACE2, that SARS-CoV-2 viruses need by looking for viral human protein interactions. All right, so the authors of our paper report that they find drugs that come from preclinical studies, clinical trials, and are FDA approved. So we wanted to take a beat and kind of break down what each of these sort of phases of drug development actually means. So whenever a lab, say at a pharma company, finds a drug that they think could be effective in treating some disease, the first step for that drug is to go into preclinical studies. So at this step, the drug is assessed for safety in animals, oftentimes mice. And basically, during these studies, scientists will take measurements like where the drug goes in the body, sort of what organs it hangs out in, how long it stays there, whether the drug gets broken down into smaller molecules that might have different effects. And once all of this data is collected, the FDA then reviews it and decides whether the drug can go into clinical trials. So clinical trials are what you hear about in the news that are like phase 1, 2, and 3. All that applies to this step in the process. And these trials take place in patients and healthy volunteers who receive either the drug or a placebo. And their purpose is to make sure that the drug is effective and safe. And then once the FDA reviews a whole other bunch of data from the clinical trials, they decide whether to approve or reject the drug. And if it's approved, then you can buy it, it goes in the market, and it can be prescribed to patients. So we have a little poll for you guys. On average, how long do you think it takes for a drug to progress from preclinical studies to FDA approval? So one end of that flow chart all the way to the other. All right, I think we're starting to level out. I'm going to close the poll in like maybe 10 seconds or so. 5, 4, 3, 2, 1. All right. So actually, ooh, this is interesting. Okay, so 3 to 5 years. I like the optimism. It's actually 10 to 12 years. So this actually kind of makes our point pretty well, and that's not, an insignificant amount of time. But that's also why this idea of repurposing drugs. Oh, sorry. Okay, maybe now you can see it. Hold on. Okay, I guess we got it. Okay, but that's why the idea of repurposing drugs is so appealing. Because if you have like a brand new drug that still has to undergo preclinical studies, all the clinical trials and everything, that can take 10 to 12 years. But if you have a drug that's already FDA approved, even though it would have to be reapproved to be used in a different disease than the one that it was originally approved for, because we already have some data from preclinical studies, we have an idea of how safe it is in people and what doses are appropriate, that process from like starting to getting it approved is a lot faster than it would be if the drug was brand new. So that's why we're trying to look for drugs that we can repurpose for COVID so we can get them into the clinic and to our patients more quickly.
All right, so this last vocab word that we're going to go over, or vocab concept, is a method that the authors use. So in this paper, the authors are trying to identify the human proteins that interact with viral proteins, as we've mentioned before. But the scientists have to figure out, how do they go from having this unknown protein, how do they identify it? And so the tool that they use to identify it, click please, is a tool known as mass spectrometry, or mass spec for short, because spectrometry is like kind of a mouthful to say. And mass spec is a tool that scientists can use to identify proteins as well as other molecules. And in this paper, it is used for the authors to be able to figure out what human proteins interact with viral proteins. Next slide. So going into a little bit more about how mass spectrometry works, spectrometry is a big word, as I mentioned, hard to say, but it really describes a method of studying a spectrum, like in this case, mass. Other types of spectrometry can look at things like color or fluorescence. And a mass spectrometer works by measuring how much mass there is in an object depending on its charge. So it basically is used to calculate the mass to charge ratio for each molecule. So So we have some videos kind of to illustrate this, because I know I'm not gonna like act it out, that would be weird. The first video is to show the first step of mass spec, which is ionization. So as you can see here, a mass spectrometer starts by breaking up a molecule, in this case a protein, into smaller bits called ions. And so now all of these smaller circles or spheres have broken up, they have to be sorted. And so these ions, or in the video, little circles, go through an electromagnet, which separates them on the basis of their mass and charge. So you can see that in this video, the little blue balls are traveling faster and the red ones are traveling slower because they're bigger. And then finally, all of these sorted ions need to be detected. And so in this step, the mass to charge ratio for each ion is graphed so that we can see how many ions of each mass to charge ratio there are in the sample. And so the results from a mass spectrometer kind of look like this piecemeal graph where you see how big each individual ion is, and then you can put that information together sort of like a puzzle to identify the protein that you put in at the beginning. And so now that we kind of have a basis of the concepts in this paper, the two questions that the authors are trying to ask are, first, what human proteins interact with the SARS-CoV-2 viral proteins? And second, are there drugs against these human proteins that can be repurposed to treat COVID-19? And how are they going to do this? They use a type of scientific reasoning that is known as experimental evaluation. There's lots of different ways that scientific knowledge can be created, lots of different approaches for that. These authors use a series of controlled experiments to understand what viral and human proteins interact and what drugs might be potentially helpful in disrupting those interactions. And so to actually perform these experiments, the authors began, next slide. Thank you. To perform these experiments, the authors began by identifying human proteins that interact with viral proteins. So they looked into the virus's genome or genetic material, and some of these viral proteins are already known, and then the others were predicted based off of what we know about coronaviruses and DNA translation in general. And so, obviously no one really wants to get those virus proteins put into their bodies to see what proteins the virus interacts with in humans. And so a way that scientists can still study human cells is to use something called a cell line. And so cell lines are cells that are originally taken from an organism, in this case humans, that are treated with a bunch of different things so that they can grow indefinitely in the lab, generally in some kind of plastic plate or something. And so most cell lines have been kept in labs for a really long time. So there are some caveats because the cells aren't exactly, they don't have all the same properties as a cell in your body would have, but they are kind of the best we have because we're not going to go around putting viral proteins into people. And so in this case, the authors chose to use a human kidney cell line, and they expressed a single viral protein at a time. And so again, this isn't exactly like what would happen during a viral infection. Normally viruses don't only infect your cells with one protein at a time, but this helps the authors to identify the specific interactions between a single viral protein and the human proteins. And so once all these human cells were expressing or they made the viral protein, they burst open the human cells and they took out all of the proteins. And they did this in a way so that all of the proteins could still be interacting with each other. So they maintained all those human protein, viral protein interactions. And then from this protein soup that they made, they pulled out the viral proteins and everything that was attached to them. And so from there, they have the viral protein connected with the human protein, and they can use mass spectrometry, which I talked about earlier, to identify the human proteins. And finally, they put all of this data together into these really pretty graphs that Em is going to talk about in much more detail, but these maps show the protein-protein interactions.
All right, so in total, of the 26 SARS-CoV-2 proteins the authors express in these human cells, they find 332 human proteins that interact with them. And then of those 332 human proteins, 66 were druggable, which means that we know of drugs that can bind to them. So then they looked at all those drugs that emerged, and you can see they come from those various phases of drug development that we talked about a little while ago. And they picked 47 of those drugs to move into viral infectivity assays. So basically, these are experiments that look at the drugs and see if they can, if they reduce the ability of SARS-CoV-2 to infect cells in any way, which we'd want to see them do if we thought they could be effective in COVID-19 patients. So to kind of break down a little bit further what their data look like. So these are two of those interaction maps that Lindsey mentioned. As you can see, there's a lot of information here, and there are actually 26 of these in the paper, one for each viral protein. So overall, this paper generated, or these authors, I should say, generated a ton of data. But just to kind of break down what we're actually looking at, so we chose sort of two viral proteins or interaction maps as examples of the data. One of the viral proteins is ORF9C, which is a protein the authors predicted to exist based on looking at the SARS-CoV-2 genome. And the other one is NSP6, which we know is involved in replicating the SARS-CoV-2 genome. So each of these viral proteins are here as the red diamonds, and then kind of at the periphery, in gray, we can see the human proteins that don't have known drugs that bind to them, at least yet. And in yellow are the proteins that do have drugs that we know can bind to them. Also pretty cool are these spokes that connect the human proteins to the viral protein. So these, based on their shading and thickness, actually give information about the statistical confidence that the authors calculated between each of these interactions. So if you look down here, this MIST score, for example, is a statistical calculation between each interaction. And then I think the coolest part of these, at least, are these sort of blue clouds that you see, kind of like in these clusters of different human proteins. So these indicate that the proteins in the cluster all share the same biological process. And those are written here in bold. So for example, these three proteins that are in this cloud are all involved in ion transport, which is basically just moving chemical ions back and forth between membranes inside of our cells. And the sort of cool thing about identifying these shared biological processes is that they give us an idea of not only the proteins that SARS-CoV-2 interacts with, but also possibly the general functions in our cells that SARS-CoV-2 is hijacking when it's infecting and replicating our cells, or in our cells. So here are the same two interaction maps that we looked at before, only now the druggable proteins are highlighted and the drugs that match them are listed in these little text boxes. So you can see of the FDA approved drugs that some of them might sound a bit familiar. For example, metformin is a drug for type 2 diabetes. Haloperidol or Haldol is an antipsychotic drug. Chloroquine, an antimalarial, is also in a relative of hydroxychloroquine. That's usually used for patients with autoimmune disorders like lupus or rheumatoid arthritis. And they've also both been in the news quite a bit over the past few months for COVID-19. So one thing to note, I should say, with these FDA approved drugs is that that's great, as we heard a little while ago. If they are effective, they can be approved for COVID-19 much faster than a drug that was just made brand new. But this also means, or I guess this can also mean that because these drugs were designed for different purposes originally and for a different disease, that they could have effects in our bodies apart from treating COVID-19, even if we take them for COVID-19. But that's the sort of thing that would be figured out in clinical trials.
All right, so now that these authors have found all of these drugs that could potentially be useful in treating the virus by disrupting viral protein-human protein interactions, they wanted to test and see if these drugs actually impacted the ability of the virus to infect cells. Again, the caveat is cells aren't people, they're not organisms, but this is a common first step that scientists use to see if it's worth spending the resources to test these drugs in more preclinical and clinical applications. And so what they did is they took cells, this time from a monkey cell line, and they began treating them with the drug of interest. And so after 2 hours, they wanted to ask the question if the drug prevents or decreases the ability of the virus to infect the cell. So after 2 hours, they infected the drug-treated cells and control cells that don't have drug treatment with the virus. And they kind of let the virus do its thing for 2 days. And the ultimate readout was looking to see if viral infection was reduced. And they did this. So the expectation is if the drug, in fact, does prevent viral infection, you would see a reduction of viral infection or maybe even no viral infection at all. Whereas if the drug didn't actually do anything, which is totally possible, then viral infection would still occur normally. And they measured the level of virus by looking at the viral proteins and the levels of viral RNA. So the authors took those 69 drugs that they identified, some of them being FDA approved, some in clinical trials, some preclinical, and they tested 47 of them in this infectivity assay to see if the drug prevented or decreased viral infection. And so this is an example of what one of those assays looks like. This is a drug called PV28. And just to walk you through this graph a little, I know there's a lot going on. The black line corresponds with the number of living cells. And this is important because we don't want the drug to kill the cells. We just want it to prevent viral infection. And the red line is the level of viral proteins. And then on the y-axis, we have percent. So the way you would read this graph is the black line corresponds to the percentage of cells that are alive, and the red line corresponds with the percentage of viral proteins in the cell. And then on the x-axis, we have the amount of drug. And so this is also important. You might imagine that giving a lot of drug to the cells might lead to the virus infecting the cells less, but it also could be more toxic to the cells and be more likely to kill them. And so in this example, the PV28 drug seems to reduce infectivity, which is awesome. You can see that by the decrease in the red line over the amount of drug. And so, but this is just one example. Not all of those 47 drugs that they tested did actually reduce infectivity. And what it would look like if a drug didn't reduce infectivity is you would have like a flat red line, just to show that there was no effect. And so of the drugs that did seem to reduce infectivity, there were two major classes, or two like groups. The first being drugs that bind the sigma-1, sigma-2 receptor, and the second being drugs that prevent making new proteins. So PV28 is actually an example of that first class of drugs that bind to the sigma-1, sigma-2 receptor. And then you would imagine it kind of like, I don't know, hand wavy kind of makes sense that drugs that prevent making new proteins might also prevent viral infection.
All right, so now that we've looked at least some representative data from the paper, let's revisit those questions that we mentioned at the beginning. So question one that the authors set out to ask in their study is what human proteins interact with SARS-CoV-2 viral proteins? And then question two, are there drugs against these human proteins that could be repurposed for COVID-19? So one thing that I think is generally kind of a good practice to take whenever you're looking at a study is not only see what the authors found out, but also what they didn't find out. You know, every paper that's out there will present a lot of data, but also leave some questions unanswered. And by kind of consciously being aware of those, you can not only see exactly how far a study pushed our knowledge forward, but also keep an eye out for the things that it didn't answer, and then for the research that's working to answer it in follow-up. So for question one, we can say the authors did identify human proteins, 332 of them, that SARS-CoV-2 proteins interact with. And they also showed evidence that some of these interactions are important to SARS-CoV-2 viral infectivity. And actually the authors' work in identifying these human proteins or SARS-CoV-2 host factors actually, well, not only points us towards drugs that might be useful in treating COVID-19 because they bind the human proteins, but they also advance our understanding about the kinds of cell functions that SARS-CoV-2 hijacks when it's in our cells. And this understanding could also help us progress towards therapy itself because although we know that, well, we have reason to think that drugs that bind these specific proteins they found might be disruptive. We can also look for drugs that disrupt these general functions that might have not been captured in this paper. And we can also test those and see if they're therapeutically viable as well. So for question two, the authors also identified drugs that might be able to disrupt the human viral protein interactions. And at least in their assays that Lindsey just showed us, some of these drugs showed antiviral activity. But very importantly, the authors did not find a treatment that we can start giving to COVID-19 patients right away. And that's because the conditions in their experiments compared to the conditions in our human bodies are so different that we can't possibly predict all of the effects that these drugs would have in a COVID-19 patient. So as Lindsey mentioned, one way in which this is the case is that all these experiments happened in cell lines. And cell lines are immortalized, as we call it, which means they can grow in a lab in some plastic, in a solution with like sugar, and just do that indefinitely and be really happy. And as you can imagine, for cells to be able to do that, they have to take on some certain behaviors that are much different than the cells in our bodies actually do. So that already makes this system quite a bit different in terms of the experimental system from what the human body would be like. Another caveat is that these cells also don't account for certain variables that would be very important to how patients respond to this therapy. So when you think about it, like patients can have all sorts of different genetic differences between them, just from one person to another. They can have other medical conditions. They can even be on other drugs to treat those medical conditions. And all of those would not be sort of factored in to the experiments that are done in these cell lines, and would also have an effect on how these patients respond to COVID-19 therapies. Now, this isn't to say that the results in this study are not useful or they're unreliable. To the contrary, I mean, just based on those two interactomes, as I said, there were 26 of those. So there was a lot of data in this paper. It came out super fast as like a testament to this power of this collaboration. And there's a lot here that'll be very useful to other researchers in the field moving forward. And this is also not to say too that the drugs they found won't necessarily be useful to treat COVID-19. It's just that we can't say that yet without actually testing them in clinical trials. So on that note, let's take a look sort of hypothetically at one of the drugs that came from this paper to see what its path might look like through the clinical trial process. So there's a lot going on here, but we'll break it down and work through it. So as I mentioned at the top of the webinar, when a drug after it's developed in the lab, and is ready to go into clinical trials, or at least hopefully. And it undergoes this preclinical testing in animals. And then once the company runs these preclinical tests, they gather all of that data and submit it to the FDA in what's called an investigational new drug application. So this application has all that data, and it also has a description of the company's plans for actually running those clinical trials. So if the FDA approves this application, then the drug gains investigational new drug status, which basically just means that it can be shipped across state lines to be used in clinical trials. So then the drug goes into these phase 1, 2, and 3, at a minimum, clinical trials. And as I mentioned before, clinical trials take place in healthy volunteers and in patients with the disease, depending upon the phase. They'll be treated with a drug or a placebo, and then their responses will be monitored over a set period of time. So clinical trials are designed to figure out two very specific things about a drug. Firstly, its efficacy, which is basically how good the drug is at treating the disease that it's supposed to treat, in terms of alleviating symptoms or healing the patient, depending on what the drug is for. And it also measures the toxicity of the drug, which means the side effects the drug has and how severe those side effects are. So in phase 1 clinical trials, which typically last about several months, the drug is tested in healthy volunteers. And here they're looking at the safety of the drug, and they're also deciding dosing ranges that would be appropriate to test in patients in the next phase. So once the drug passes phase 1, then it goes into phase 2, and now it's given to patients with the disease the drug is actually designed to treat. So here we have quite a few more participants in this phase and in the previous one. And it also lasts a bit longer, going from several months, like up to a year. And then following phase 2, then the drug moves into phase 3 clinical trials, which is still in patients that have the disease that the drug is meant to treat, but now there are way more of them, on the order of hundreds to thousands. And this phase also takes quite a bit longer, usually in the realm of 1 to 4 years. So here, as in phase 2, they're still looking at the drug's efficacy, at its toxicity, and kind of narrowing down doses that would be appropriate to use in the clinic once the drug is approved. But in phase 3 in particular, they're also looking for rare side effects that you'll be less likely to find when you're testing the drug in fewer people as you are in phase 2. So when you see drug commercials and at the very end, the narrator kind of like whispers extra side effects at you like a secret, a lot of those show up in phase 3 clinical trials. So after all the clinical trial data is gathered and the trials are done, and the company packages that up into what's called a new drug application. They're usually at least 100,000 pages long, so it's a lot of data. And the FDA reviews that, and then at that point either approves or rejects the drug, at which point it can be marketed and it can be used to treat patients. So as I mentioned before, FDA approved drugs that are approved for one disease must be reapproved before they can be used to treat a different disease. So now to look at one of our hypothetical drugs. PB28 is the one that Lindsey showed us some infectivity data for. That looked really good. But because it's not an approved drug, it's actually quite far at the very beginning of the pipeline. It would be entering here in the preclinical phase. And as we learned at the beginning, that means it could be like 10 to 12 years between this drug actually entering the pipeline and coming out at the other end and being FDA approved, if it makes it through the pipeline at all. By contrast, remdesivir, which is a drug for COVID-19, that's being tested for COVID-19, that's been in the news quite a bit lately, was developed as a general antiviral. It was actually in phase 2 clinical trials back in 2015 during the Ebola outbreak, and entered phase 3 clinical trials this past February in a trial that included some COVID-19 patients, and it's still in that phase of trials right now. And actually, there's kind of a special sort of caveat with remdesivir. So as of last week, because of some preliminary results that suggest that it might be shortening the duration of disease in certain patients, and the fact that it's just looking kind of promising, the FDA granted what's called emergency use authorization for remdesivir, which means that even though it's not FDA approved yet, doctors can prescribe it to patients that have severe symptoms. So as I mentioned, like this rule about approving drugs for a new disease still holds true, but given the promise of this drug and the special circumstances that we're in with COVID-19, there are some exceptions that can be made so that way these drugs that look promising can get to patients that they can help even sooner. All right, and one last thing that I want to share here. So resources that I found are pretty helpful and kind of just looking into, a bunch of COVID-19 clinical trials that are going on and sort of preparing for this data for the people presentation, I've listed here. So statnews.com has a really nice COVID-19 clinical trial tracker. It's very reader friendly, really up to date, and has like nice descriptions of what the drugs actually do and how they actually work. The FDA, of course, has great information about how these clinical trials are happening. They also issue frequent press releases with new findings, like that emergency use authorization that I talked about. And lastly, if you want to see all of the COVID-19 trials going on all over the world, those are available at clinicaltrials.gov, and they actually have them from the WHO database. That's the fullest list.
All right, so we just wanted to end this presentation by highlighting how this was a totally massive amount of data, and the reason why the authors were able to generate all this data is because of the fact that there were 125 authors in over 20 labs in 8 institutions. And we also just thought that it was super nice that in addition to the fact that there's already this massive collaboration, the authors have also done a really good job of making all parts of their research as accessible and transparent as possible, including writing in the paper what their next step plans are for testing these drugs. And they're also really willing to distribute materials to other labs so that other labs can participate and help testing. So we wanted to highlight this by one of the quotes from the paper that just like made us all feel a little happy and kind of optimistic in these very uncertain times. And this quote says, "Given the urgent and acute global need for SARS-CoV-2 research, we will make these resources available to the scientific community upon completion, as we have done with this study." And so we just thought that was really cool. And it's nice to know that scientists are all collaborating and working together to try to figure this out.